Hi class, I am the Microplitus crocypes wasp. Today we're going to learn about my interaction with the tomato plant and the Helicoverpa caterpillar. I live a unique life as I am friends with the tomato plant, but the caterpillar is my enemy. I hope you enjoy the research that's being conducted on me in this laboratory located on campus.
Why would a mutualistic relationship between a plant and a wasp require a caterpillar to expel the wasp's larvae? This bizarre relationship is what Gary Felton and Michelle Pfeiffer have been investigating for more than 15 years in their research lab located in the Agricultural Sciences Building at Penn State University. This interaction is what they call a three-way dance between a wasp, tomato plant, and a caterpillar. This three-way relationship covers two of the different types of symbioses found within the coaction compass. Specifically, predation is seen between the caterpillar and wasp, and an indirect mutualism can also be seen between the wasp and the plant. Not found within the coaction compass is the herbivory interaction between the caterpillar and plant.
This is the Helicoverpa, a caterpillar with a wide host range being studied in this lab on tomato plants. As the caterpillar eats the tomato plant, it releases a chemical called volocitin found within the saliva. As a defense against the caterpillar, the plant releases airborne chemicals known as monoterpene, sesquiterpene, and indole that attract the Microplitus crocypes wasp. Here, you can see the reproductive organ of the wasp dissected. The wasp completes its life cycle by laying its eggs within the caterpillar, as seen here, while also injecting a virus known as MCPDV. Compared to the several larvae expelling from the first caterpillar, however, this specific caterpillar-wasp interaction can only develop one larva per relationship. This virus does not reproduce, but instead expresses some viral proteins that inhibit the caterpillar's immune system. This allowed the wasp larva to develop. Approximately 10 days after the parasitization, the odd phenomenon where the wasp larva emerges through the caterpillar exoskeleton to form a cocoon occurs, eventually allowing an adult wasp to develop. Once the wasp pupates, the caterpillar dies, therefore having a positive effect for the wasp and a negative effect for the caterpillar, resulting in a parasitic relationship.
The caterpillars are cannibalistic, so they eat each other. So they need to be reared individually, or when they're very small, we can put like 2 or 3 in a cup. But as they get bigger, we can only have 1 in a cup, which is why we have all these little cups. Each one contains a caterpillar. And what Ching Wen is doing right now is pulling out small caterpillars and putting them in the petri dishes. And then she'll put a wasp in there as well so that it can lay its eggs in the caterpillar, and that's how we maintain the colonies of our wasps. So in addition to the experiments that she's doing, she also spends a lot of time just maintaining the 2 species of insects. And then in the greenhouse across the street, we have all the tomato plants that are growing as well.
As previously stated, through this three-way dance, an indirect mutualism can also be observed between the wasp and the plant. The plant induces the caterpillar to release chemicals that attract the wasps, preventing the caterpillar from consuming the plant. In return, the wasp is given a way to lay eggs, completing its reproduction cycle. This positive relationship represents the second of 3 relationships within this research. We will now visit the lab where Marissa is interviewing the lab technician, Michelle Pfeiffer.
So thank you, Michelle, for meeting with us today. We wanted to do an interview to kind of explain to the class a little bit more about the project and what's going on inside the research lab. So first, we wanted to kind of get a better idea of what are the project goals.
So the project goals with this are to better understand the relationship between the wasps and the caterpillars and how the plant reacts to the parasitized caterpillars versus non-parasitized caterpillars.
And so what are the applications broadly of this research?
The caterpillar that we're working with, the corn earworm, is a major pest on plants throughout the entire U.S. and it has a huge host range and causes a lot of economic damage. The wasp is a way to help control the caterpillar because the parasitized caterpillars die. So by better understanding this relationship, we can develop new and better control methods and reduce our use of pesticides to control this caterpillar.
So what is your favorite aspect of doing the research here at Penn State?
I like the fact that we're doing different things every day. It's not repetitious at all, new exciting experiments, and we're learning things that people have never done or seen before.
So how did you specifically end up here at Penn State doing this research?
My degree is actually in plant science from Penn State, and so I've been working as a technician most of the time, and when Gary Felton came to the university, he was looking for a technician, and so I started working for him then.
And so how long have you been studying relationships now?
I've been in this particular lab for 15 years.
And have you guys looked at a couple of different plant caterpillar relationships? Or?
Yes, so we study plant herbivore interactions, and we mainly work with caterpillars and tomato plants, but we've also done a lot of work with corn. We've worked with some beetles, the wasps, some bacteria, and we have a lot of different projects going on at any given time.
And so if undergrads are interested in getting involved more, are there ways for them to get into the research lab?
Yes, we have undergraduate independent study projects. We have 2 undergraduates currently working in the lab, and occasionally we hire undergraduates as wage payroll during the summer as well.
Well, thank you for taking the time to show us around the lab and to give our class a better understanding of the three-way dance or the different relationships.
You're welcome.
As this is a fairly new project, no papers have been published on the research. However, this team recently received a grant from NSF to continue working. Within this continuation, they plan to answer questions such as which viral proteins are expressed in the caterpillar and how they affect the caterpillar's immune response, and does the plant respond differently to parasitized versus non-parasitized caterpillars. We hope you enjoyed this tour through this unique Penn State research lab.